Hey guys, welcome back to my channel. So moving forward with today's topic, that is pharmacokinetics. So what is pharmacokinetics? So basically, activity of drug inside our body after we take it is simply known as pharmacokinetics. The movement of the drug throughout the body, and also it includes the onset of action, duration of action, and intensity of the drug effect. All these comes under pharmacokinetics. So our body has a normal tendency of not allowing any foreign molecule inside our body. So for the drug to reach to its target and show its therapeutic effect, the drug molecule must cross a number of restrictions. So the drug must be absorbed, and then it should be distributed by crossing the membrane barriers, and it also have to survive the metabolism and elimination before it has shown its effect. And then after it has shown its effect, it can just get eliminated and metabolized also. So the processes that comes under pharmacokinetics are absorption, distribution, metabolism, and elimination. So starting with today's topic, that is absorption and mechanism of drug absorption, that is how the drug is absorbed. Starting with the first mechanism that we have, it follows is passive diffusion, then it follows facilitated diffusion, active transportation, endocytosis, and filtration. So we will see what happens in case of passive diffusion. So in case of passive diffusion, there is a higher concentration and there is a lower concentration, and we have our cell membrane. So this cell membrane is lipophilic in nature by outside. So what happens when we have our drugs that are uh, hydrophilic in nature, or they can be lipophilic in nature? So in case of hydrophilic drug, they can simply just cross through the pores and they will come inside the cell. They will diffuse easily. And in case of the lipophilic drug, they will cross the barrier through the membrane itself. So in case of passive diffusion, we have seen that there is no carrier requirement. That means any drug can easily diffuse from higher concentration to the lower concentration without the help of any carrier. And they also is energy independent, that is they do not require any kind of energy. Now in this case, since there is no involvement of any carrier, therefore they cannot get saturated. They won't get saturated actually, because the number of particles or the number of solute that are present, they can easily get diffused, irrespective of the carrier or anything that are present over here. And they are also not specific for any particular structure. That means whatever the structure be, they can just simply go into the cell. So this was about the passive diffusion. Also, most of the drug, what they do is they follow this pathway, that is our passive diffusion. Next, moving into another mechanism, that is we have facilitated diffusion. So in case of facilitated diffusion, we have carriers. So these carriers, they are present within the cell membrane itself. So we have our drug that will come and it will get bind to these carriers. So as soon as the drug come and bind to this carrier, there will be a conformational change. So after there is a conformational change, this drug will get inside the cell by the help of these carriers. So there is an involvement of the carrier in this process, but they are also energy independent. They do not require energy. They are just driven by the concentration gradient. That is, they move from higher concentration to the lower concentration. But in this case, since there is involvement of the carriers, so it may happen that after some time they can get saturated. Like if there is a situation where the concentration of drug is more than the number of carriers, then all the carriers will just get saturated with the drug, and there will be a time where some of the drug will remain uncombined with these carriers. So a time will come when all the carrier will get ultimately saturated, so they are saturable. Also, there can be a competitive inhibition, that is, if there is any drug molecule or there is any enzyme or substrate that are similar to the structure, then they can compete with them and they can ultimately bind to these carriers. So also competitive inhibition can occur in this case, that is in case of facilitated diffusion. So this process is known as facilitated diffusion. Then third comes the active transport. In case of active transport also, there will be the function of the carriers. So carriers are as usual present in this case. So whenever we have our drugs, they will go and they will combine with the carriers. So after they are combined, there will be conformational change, and after there is conformational change, the drug will get into the cell. So all these processes are energy dependent. And from where do they get the energy? So energy they will get by hydrolysis of ATP. So this ATP will help them to carry out all these reactions that are being carried out. So one thing in this case, in case of active transport, they can move the solute particle from lower concentration to higher concentration. In case of passive diffusion, in case of facilitated diffusion, we have seen that they move the solute particle from higher concentration to the lower concentration. But in this case, they can go uh, they can go from lower concentration to higher concentration. Also, the same problem that we have seen in case of facilitated diffusion goes with active transport, that is, they can get saturated. Also, they are very selective in nature, since they have some conformation produced over here. So the drugs that will match their position at this place will only get into the cell. Also, because of this, there can be competitive inhibition. So this was about active transportation. Then comes endocytosis. So endocytosis is usually for the large molecular particle. So when the large molecular particle goes and combines with the protein binding site which is present in the cell membrane, they will get into the cell. So they will form a vesicle. So the drug is entrapped in the vesicle. This vesicle will carry the drug to the other side of the membrane and will fuse over this part. So as soon as they get fused here, all the content or our drug, it will get into the extracellular space and ultimately they will be released. So this process where it is getting released is known as exocytosis. So where they are getting engulfed, it is called endocytosis, and the part where they are getting out of the cell is known as exocytosis. So this is particularly for the larger molecule which cannot fit into the pores or cannot come through the channels. So the rate of absorption is dependent upon few factors, that is aqueous solubility, concentration, the area of the absorbing surface, the route of administration, and the blood circulation over that place. So these are the factors that are responsible for increasing the absorption. So this was it for today's topic, that is absorption and the mechanism of absorption. In the next video, we will see the other part of pharmacokinetics, that is metabolism, elimination, and distribution. So thank you.